Welcome to everyone joining. It is noon by both my phone and my computer. So welcome to today's webinar, IPM Strategies for Reducing Insect Pest Damage in Midwest Blueberries. My name is Laura Isles. I'm one of the co-directors of the North Central IPM Center. We're one of 4 regional IPM centers supported by USDA NIFA Crop Protection and Pest Management Regional Coordination Program. We have a mission to coordinate IPM across the region by sharing and promoting IPM research, extension, and teaching activities. If during today's webinar you have any questions, please type it in the Q&A part. You should see it at the bottom of your Zoom screen. And then after the webinar, I will read off those questions to our speaker and they will answer the questions then. So I am excited today to introduce Rufus Isaacs from Michigan State University to update us all on spotted-winged drosophila and blueberries. Take it away, Rufus.
Thanks. Yeah, thanks, Laura, for the introduction, and great to be part of this Pests in Progress session. I think we're going to talk about one particular pest today, and then hopefully some of the progress we've been making on trying to work out ways to manage spotted-winged drosophila. This grant is one of the ones that was funded by the Crop Protection and Pest Management Program, so I'm going to focus on that, but there's also a broader view of some of the work that's going on around the country with spotted-winged drosophila. So yeah, thanks to the North Central IPM program for setting this up, and hopefully we'll get a little time at the end for questions. I did want to start with a photo from one of our growers that we collaborate with. This is a farm in Southwest Michigan, and it just gives you some of the sense of the large-scale blueberry production that we have in Michigan. As an industry that's faced spotted-winged drosophila, we've been doing a lot of work to try and help these farmers be able to harvest fruit that are free of that pest. But it does also highlight just the volume and scale of some of our blueberry production here in Michigan. But I do think what I talk about today will be relevant across the region, wherever you're growing maybe blueberries, but also raspberries, blackberries, some of the other affected crops. I think it's good at the beginning just to spend a minute talking about how the landscape of pest management is changing. We know that this is a continual change. Pest management is never static, but specifically within the blueberries system and the berry crop world, we're seeing much greater quality expectations from the marketplace. You go to the store and people want the perfect, large, beautiful looking fruit. The breeders are doing a lot of work to sort of catch up with that demand and developing new varieties that will meet the market expectations. So there's some change of cultivars going on. And the climate is changing too. We're seeing changes that affect certainly the pests and probably the crops too, with warmer winters, really different spring weather, and some hotter summers. And 2025 is a good example of that. There's always these new regulations on pesticides that change what we can and can't use. And then on top of all that, we're getting these invasive species like spotted-winged drosophila that can really turn our IPM programs upside down. And that's exactly what this insect did when it came into the country in 2010, around that time. Hard to believe it's now almost 15 years ago that we really started realizing that this was a challenge. I think we have made progress, and I'll talk about that today. Not maybe as much as everybody would like, but definitely some things to share with this group. And today I'm going to focus on this one project funded by the CPPM program, and it's a collaboration between me and some colleagues at Michigan State University, Hannah Levinson and Jim Walgenbach at North Carolina State University, and then Vaughn Walton in Oregon. And we propose these 4 objectives, and I'll talk about most of these as I go through today. So you'll see pieces of the work we've done to address those 4. And this is part of a bigger national effort, certainly not just these 3 universities. I think every group that is working in berry crops or cherries, some of the affected crops, they're focused to some extent on spotted-winged drosophila, and a lot of that information is encapsulated in that SWD management website that I put at the bottom there. So if you're looking at this recording and interested in some of the other work that's been done on this pest, definitely check out that website. Okay, so in Michigan, focused on blueberries and focused on insect pests, we deal with a sequence of insects that challenge blueberry producers from the early spring with cutworms as things warm up in March or April, all the way through to bud mites that might be moving to overwintering buds right now in this time of the season. And the ones that are in blue here are the major economic pests. So that's what drives most of the spray applications. We used to be able to get away with maybe 2 or 3 applications of insecticides to protect from blueberry maggot in the harvest season. And now with a much more quickly reproducing and damaging pest that can build up its numbers quickly, spotted wing Drosophila has kind of increased that pesticide use. So we're trying to develop ways in an IPM context that we can reduce the need for as many insecticide inputs. Obviously, that should help reduce the development of resistance, and we haven't seen resistance in Michigan and other Midwest areas in the testing we've been done, but out in California, there are areas where there's populations of spotted wing Drosophila that become resistant to pesticides. So we're trying to find ways that we can minimize that selection pressure and hopefully keep being able to manage this pest. All right, so let's talk a little bit about spotted wing Drosophila. It's not just in this region, it's around the world, and it's become a pest because as it's moved from Asia into these other areas, it's been able to, unlike the other flies, it can lay its eggs into ripening fruit. So that puts a larva there potentially at the time of harvest, and they can build up their numbers really quickly. There is some monitoring technology that's been developed. You can purchase or make your own traps and put baits in them. We'll talk about that in a minute. And those are really good at giving you some early warning that the flies have become active and are out there, but they don't tell you a lot about what's happening in the fruit, the actual risk to the berries, which is what we're more interested in. So I'm going to talk today about a way that you could monitor fruit for perhaps eggs, these little eggs with the tubes on them, but those are very small. I think it's more realistic to be able to sample fruit and look for these very early instar or stage larvae and make decisions about whether you need to protect them from those, control those, so that you never get fruit with these much larger, easier to see insects in them. During this time of the season, in this part of the world, we also have blueberry maggot to deal with. So I'll talk about that for part of the presentation. And then I'm going to update this audience on the biological control work that's underway to try and spread biological control wasps. These are tiny little wasps that lay their eggs inside spotted wing Drosophila larvae, and kind of talk about where that's at in the research phase at the moment. So blueberry maggot, let's go back to this insect. As I said, it's been around forever, but we've seen so much pest management focus on spotted wing Drosophila that I like to remind people of what this looks like, because it's might have sort of taken our eye off the ball a little bit here with blueberry maggot. It's a fly that overwinters in the soil, typically comes up in our region to be active in the 3rd week of June. And we monitor it with these yellow sticky boards that have a bait on them. You can get the smell in the glue, or you can add the smell with these little plastic containers. They're available from Gemplers. I think Great Lakes IPM sells them. So you can monitor those once a week and look for the fly that has the wing pattern on it that I like to think is an M. You have to use a bit of creativity there. But if you think about this as an M, and remember maggot or MSU, that's a way to remember that pattern. And then if you see those insects, you've got about a week to protect the fruit. And so we typically would apply about every 14 days one of these effective pesticides that could protect the fruit from blueberry maggot. There's an organic option, and then lots of other kinds of insecticides that could work effectively against this insect. So that's how this was managed. Typically, we have a degree day model that's up on our EnviroWeather site here at MSU to sort of prepare for putting the traps out. And it's a fairly manageable pest. But then along comes spotted wing drosophila, and as I said earlier, it kind of turns it upside down, because if you only were to protect the fruit every 2 weeks, if that insect was active, you really would find it hard to keep up with its reproduction. So at first we used put a lot of effort into trapping with these container traps. This is a homemade one with some yeast and sugar in the bottom, and that's very attractive. Flies go in the little holes. The small holes also stop you getting a bunch of moths and other things in there. And this can tell you when the fly is active, can tell you when the surge happens. Typically, there's a part of the year where the population just really picks up. But it doesn't tell you, unfortunately, what's happening in the fruit. It just tells you that the flies are active. And we've had situations where there's infestation happening in the berries and low catches in the traps. I think because the fruit are just much more attractive than the trap. It's also kind of messy if you're dealing with this version. There is now a synthetic lure that, again, you can buy from some of these trap supply companies. There's a synthetic lure that you can use with soapy water in the bottom, and that is a lot cleaner to use. But again, still doesn't relate directly to what's happening in the fruit. Some groups have looked at egg monitoring. As you can imagine, that's the surface of a blueberry with a tiny little tube that's stuck out from the egg. As you can imagine, that is very time-consuming and difficult to find low levels of these eggs in the fruit. It can be done, but we've found most growers and consultants don't really have the time and patience for that. So we've been focusing more on this larval monitoring method, where you pick some fruit, you can put it in a salty solution. Larvae, if they're there, will come out of the berries, and you can collect those in the filter. This is a little coffee filter, and I'll show you some details of that in a minute. This is similar to the old boil test, where growers used to take fruit, put it in some water, boil it for a minute, usually using a microwave or a stove, and then the larvae would float. This doesn't require any equipment, really sophisticated equipment like that. So it can be done in the field, in the back of a truck, as you're going around to multiple farms or fields. So we're trying now to combine that method with some decision making, trying to think about where are the opportunities to reduce pesticide use, where can we use this to help guide decisions that are a bit more based on the reality of what's happening in the field rather than what we fear might be happening. And this method can also provide some confidence in your IPM program, because if you sample and you get experience with it, you might be then able to use it to say, I'm confident that my fruit don't have infestation and that my spray program has been working, or my management program has been working. And it can also help with harvest scheduling, just because you know that your fruit can be picked, can be marketed, and you won't run into problems with contamination. So here's a quick run through this filter sampling method. This is a research setting, so we use gloves and maybe a bit more formal than it might be in the back of a truck. But we take a sample of, say, 4 to 8 ounces of fruit, so like half of one of these little containers. Give it a quick squeeze, put it in water. It can be salty water if you want to kill the larvae. If you just use plain tap water, they'll still be wriggling, but you can get them out of the berries still. And then we use a setup that's some hardware cloth in a funnel, and that coffee filter is just underneath there. So give it a quick rinse, and you can, if you've got multiple fields, you could have them all labeled by field, by cultivar. Now, I'm not suggesting everybody does it at this scale, but this is what we use for our research trials, and we can do maybe hundreds is a bit of an exaggeration. We can do tens of these in a day. Lots of different coffee filters. We found this one in the top middle to be really good. That's like $6 at Amazon.com. And I've got a guide. If anyone wants to email me afterwards, that'll be my email address. I've got a guide of where you can get all this equipment, and it's pretty straightforward. but we've got some plans for how to like build the filter and where to get the coffee filter from. So you end up with a coffee filter where if there were larvae in the bottom of that sample that came out of the berries, you can see the eggs, you see the first, second, and hopefully not, but the larger third instars. This is SWD spotted wing on the bottom right, and this is the shape of a blueberry maggot larva. So I hope you don't ever see any of these, but if you do see this more stubby ended, almost like a carrot shaped larva, but they're white, that's a blueberry maggot. And so you can get information on both from your sample. This is what it looks like if you've got a hand lens or really good eyesight or even a microscope. Just takes a couple of minutes to go through each sample. And you can see here I've ringed in red the larvae that are present. This was an untreated plot late in the season. We picked these fruit, and within an hour we knew that they were infested with spotted wing drosophila. Okay, so you get that information and it tells you something. If this was the situation, it would tell you that you absolutely need to take some corrective action. You might even decide to, I've seen growers do a picking and not market that picking and then come back in after it and clean the field, protect the field from future infestation, and then keep sampling and know that next harvest will be good. Here's something graphical that we laid out, and I'll kind of walk you through it here. Where we sample the berries every 3 to 7 days, depending on your cycle of picking and other activities that you're doing. But if you do this sample and you find no SWD, you do not need to spray. If you find low levels of SWD, so like maybe let's say 1 to 5 in your sample, if harvest is coming up soon, you might still not spray because there's very low density and maybe they're the tiniest little larvae. And as soon as you pick, you're going to cool them down to stop any further development. But if you've got, say, a week left, you might treat that field with a residual pesticide. And then, of course, if you go in and you see high SWD infestation in that sample, you might still be treating, but thinking more about the pre-harvest interval and being able to get back in there in time for your planned harvest date. So this is pretty simple, but what we're hoping is that it can at least start giving farmers, berry farmers, some confidence that they could make this decision over here and not have to just do a calendar application every week. So just to review the uses for this, it can give you some sense of when to start applications, and I'll come back to that when I talk about the last few years of experience with this. We also get rain in Michigan, as I'm sure you do in your region. And if you've treated and you've still got 5 days till harvest and it rains 2 inches, you can go out and do another sample and see where you are and see whether you're going to make a decision to treat, to protect, or wait until harvest time and then maybe reapply after that. So it can help in that kind of cycle through the harvest season. And then, yeah, I think it's important just to mention that it can help confirm that your system is still working. I've emphasized how this kind of filter test for spotted wing can influence your decisions to save sprays. And it certainly did that for the farms that we worked with in 2023 and 2025 on this project that I'm highlighting today. So in those 2 years, we had hot, dry spring weather. It was very unsuitable conditions for spotted wing drosophila. It also wasn't much fun to be in the field in some of those days for humans, but they don't like it when it's hot and dry. And so those conditions really pushed back the increase in that SWD population later in the year, later in the summer. And so if you grow blueberries, you might know the cultivars blue crop, for example. We were able to pick the first and second picking of blue crop in these farms without pesticide application, insecticide specifically for SWD. And then once we got to the later part of blue crop, protect that and the subsequent later varieties. In contrast, 2024, we had a really mild winter the year before, and then we had a cooler spring, and it was really perfect for SWD. That year, we were doing these samples, and almost as soon as we started sampling, the samples, the information coming back from these very simple filter tests were telling us that SWD was already active. We even did some sampling in the wild fruits like the honeysuckle and other things, raspberries around in the woods. And that gave us some early warning that we really needed to protect the fruit. And so that's another side of this coin, that it's not always saving sprays. Sometimes it might save the crop, so you're not caught unawares. Here's a couple of like just graphical examples I'll run through from a couple of farms where I think this was from 2023. But we did the farmer did one insecticide application just when the fruit started to ripen because they wanted to protect from blueberry maggot, and they'd had some issues with that in the past. So on July 7th, we went in and checked all of the SWD samples. These are fruit samples that were done in all these locations. 0 larvae. So we skipped the insecticide sprays that week. July 12th, still zeros. And then on the 18th, there was one larva found, and that grower was very risk averse and said, okay, you've got one larva, I don't want any more. So they protected the whole farm, and subsequent weeks just kept that spray program going. But they had saved a couple of pesticides early on in the season. This is a different farm, obviously, different layout, and we did the samples in these different areas. They skipped an insecticide application in early July, and the same in July the 12th. But then we checked for blueberry maggot in some traps that were on that south side of the farm near some woods. And even though they weren't finding any spotted wing drosophila, they put an application on for blueberry maggot, which would have also protected from spotted wing drosophila. So then they just kept that program going as the pest developed and the fruit ripened. So it's obviously not a silver bullet, not a complete solution to this, but it is, I think, a tool that we can start to add some sampling and thresholds into blueberry management. Then the question comes, well, what should I spray if you do decide to treat the field? And this is some data that was compiled from a whole bunch of entomologists. It's not all from people that had tested every single pesticide on the graph. So it's more like, would they rank these products as being weak, fair, good, or excellent? And so I would just say the products that you see that are above good would be the ones to be thinking about. They're not all registered in all berry crops, so I think that's something to keep in mind. If you grow raspberries or blackberries or cherries, may not be the same as if it's in blueberries. But we do emphasize the need to rotate chemical classes. So this is it colored by chemical class. We have a bunch of pyrethroids, so I like to pick those out because Mustang Max, Danitol, Asana, Brigade, Hero, Baythroid, they're all the same chemical class. So you don't want to rotate between those. They tend to get used a lot because they're more cost effective, but I think rotating with one of these other classes would be, one or more of these other classes would be a good plan. So just to wrap up the larval sampling part, talked about this, these points, but it is a tool that we're starting to use and starting to teach farmers through this project. And maybe it's not for every farmer to use, but maybe their crop consultant could be using this and help them save some money and maybe save some of the sprays. This is a little bit of what I described earlier with the wild host plants. So this is a honeysuckle type of plant that we picked samples of. And 2025, again, as I said, was one where there was hot, dry conditions, and that really delayed SWD development. So from year to year, as our climate varies from season to season, this is a tool that I think we can use to adjust spray programs. All right, I promised you I'd spend a little time on the biological control program for spotted wing Drosophila. This is a slide with a couple of photographs, very up close photographs, because these are probably a millimeter or two along between the head and the end of the abdomen. Tiny little insects that lay their eggs inside the fruit to get inside the larva. So you can see on this photograph here, there's just a little hypodermic needle there that it lays its egg inside the fruit. This one on the left has been approved for release by USDA after extensive testing. This one on the right did not get approved, but we'll talk about that one as well during this section. So here's what it's doing. This is a photograph from a laboratory setup, but this is some diet that we've got larvae developing, and there's one right here that you can see. So its tail is over here, its head is over here, and that wasp is laying its egg inside that larva. A lot of our native species of wasps like this can basically suffocate the egg. So the wasp lays the egg in the larva, but it gets suffocated by the defense mechanism that the larva has. And that egg turns black and can actually end up in the adult, but it never turns into a wasp. So that wasp didn't get a chance to kill that fly larva. These species from Asia that have been evaluated are resistant to this defense mechanism. So they have much greater chance of, instead of having a larva develop out of your fruit and more and more and more of those as they reproduce really well, instead of that, we get a wasp that can then, again, go around and look for that larva inside fruit, lay its egg, and then we get more and more wasps instead of the flies. So that's this phase we're at the moment, trying to determine how well can these wasps suppress spotted wing Drosophila and do that in different parts of the country and around the world. We've been able to get the permits to raise these at Michigan State, and other universities have done that too, and other USDA labs. Here's just a photograph of us releasing them from a trap in the field. And they're tiny little things, come out, and if you watch these, you'll see them very quickly go and find a fruit and start evaluating that fruit to see if it has a larva in it, and then move on to the next one if it doesn't. So they're like heat-seeking wasps out there looking for their host. There's been a big effort through funding from USDA to release these in different states around the country. 2022, there were over 50,000 of these released. And you can see the numbers there in other years. And you'll notice the L. japonica was that second species. In 2022, 23, we started finding that in other parts of the country, even though it hadn't been approved for release. So this species seems to have hitchhiked here. And if I updated this map now, you'd probably see those blue dots in most of the areas where people are active studying this, because we've been looking so much for the other species that we found this second one. The second one seems to be doing very well. It's in most of these areas where fruit's produced. It's getting on to spotted wing Drosophila, and we're rearing it out of a bunch of different host plants. So it's in the blueberry in the fields that we have been monitoring, but we're also finding it on all these other host plants that you see the blue bars on. So that means it could persist out in the woods, have a reservoir out there, and come into the farms when it's needed. Kind of shows it here where spotted wing Drosophila picked up in June and then into July, and this Ronin wasp just built its numbers through the summer. So we're hopeful. We don't really have a final answer yet on that and its influence on spotted wing populations. But it's great that we have that here, and we're hoping it will take off that peak when SWD really builds up late in the season. I'm going to wrap it up here with a couple of just final slides to reiterate, I think, the importance of blueberry maggot management. If you're in areas where that's been a pest, that can also be monitored with the filter test. Spotted wing Drosophila monitoring, I think, is something we're really trying to emphasize for IPM programs in crops that are affected by spotted wing Drosophila. Can definitely improve decision making there. I didn't focus on it today because of time, but we're also doing some work on like physical ways to try and make farms less suitable to SWD. And one thing that's showing some promise is this mylar. It's like a reflective mulch. Plants could be planted into it if you set the barrier out in advance, or you could put it in retroactively like we did for this research. But with the ultraviolet light that gets reflected back up into the canopy, this reflective mulch is both reducing suitability for spotted-winged Drosophila, reducing infestation, and then my horticultural colleagues are measuring the effects on photosynthesis, and it's definitely also having some horticultural benefits to make the plants grow a little bit better and also ripen the fruit more evenly because you're getting light kind of scattered up in the canopy from different sides. So that's an interesting thing we could talk about later if there's time in the questions. And then, yeah, just to, again, say what I highlighted on the biological control. There's some work being done on releases, and then we're monitoring to figure out how much and where we're getting the best levels of suppression of SWD from the biocontrols. So I just want to end by thanking this group for a chance to talk to you all. This is my team here, photographed a couple of years ago at Michigan State. We were funded for this specific project by USDA NIFA's crop pest management program, and also by our Michigan blueberry growers. So thanks to them and the Horticultural Society for supporting some of this with funding and partnering with Michigan Department of Agriculture. If you have questions, I think there's a bit of time at the end here for questions, but that's my email address, and I wish you a relaxing winter while the plants are also relaxing, and hopefully some of this will be useful for your pest management in 2026.
Thank you so much. I didn't realize that blueberry stems were kind of reddish like this.
Yeah, it looks great in the winter. And this wasn't a particularly sunny day, but if you get a bright sunny day with the white snow and the red blueberry wood, it looks really great.
Very good. Oh, and if you can just swap to the QR code, so anyone who needs that can get that information. And then if you have any questions, please pop that into the Q&A box down at the bottom. The question I have, I know how devastating spotted-winged Drosophila was when it came in. It does seem longer than 15 years. I know that I haven't paid attention in years. Is there a sense of like what they're on earlier in the season, or how are they moving to different crops, or like what does that kind of look like overall?
Yeah, so they have a winter as mature mated mature adults, which is kind of interesting in itself that they can make it through very cold winters. I think the fact that I just showed that snow photograph, I think in our region there under that blanket, that helps. And then, yeah, as it warms up, there's some studies showing that even before there's berries out there, they're persisting on mushrooms and sap, and just being really opportunistic on anything they can get to get a little bit of sustenance on a warm spring day. And then as soon as there's strawberries, that's probably the first, June bearing harvested crop in the springtime. And if there's any wild berries that soften up, as soon as those get some level of softness, they'll be trying to lay eggs into those. So we tend to see very low levels for months, all the way through May, June, July. And then it's often as we get into August when there's a lot of ripe fruit in the landscape, whether that's wild or commercially produced fruit, and the weather conditions are warmer and more suitable, they will then pick up their reproduction. And it only takes them 10 to 15 days to do a life cycle, and they can lay a couple of hundred eggs per female. So you can imagine the reproduction can quickly pick up.
Wow. So I know you had the kind of the map of the blueberries, but how long is kind of the harvest time of blueberries, and does the SWD pressure stay the same or increase?
Yeah, here, and it would adjust maybe for different parts of the country, but for here, we start picking right at the end of June, and a farm that started in early June would probably be done by mid to late August. But we also have areas further north in Michigan that would start later and might still be picking into September. So yeah, so it can be multiple weeks, and growers have multiple cultivars from very early to very late ripening ones that helps spread out that harvest season. But they all sort of overlap, so then they can have their picking crews and the harvest machines running through that harvest cycle.
Okay. So they could be, so yeah, I hadn't realized it was so long. So they could be having to apply a lot of treatments for SWD without the monitoring.
Yeah, maybe not to the same field, but yeah, if you had adjacent fields with different timings of harvest, typically each field's getting picked maybe 3 times on average. So that would be like 3 or 4 weeks of harvest in one field, and they would need to protect that ripening period to make sure it's free of SWD larvae.
Okay. And if anyone else has questions, please type that into the Q&A. I just have one on the synthetic lure you mentioned. Is that one specific to spotted-winged Drosophila, or is it attractive to all fruit fly?
Yeah, unfortunately, we don't have a species-specific lure. So you do to use the trap, and I didn't mention this, but it's good that you brought it up. To use that trap accurately, you really have to be able to determine spotted-winged Drosophila from the other vinegar flies that will show up. So we've got 2 or 3 other common species that tend to be in those traps in this region. So luckily the males have the spots on the wings, so that's easy, but the females don't. So, you know, we're typically emptying out that trap, filtering the trap contents. It's easier if you use the clear liquid, but yeah, filtering the liquid out and then going through that either with a hand lens or under a microscope.
And growers and stuff are pretty good at picking them out and stuff at this point?
I think it depends on the person and how much time they have and how much experience they have. It's not easy, but it definitely gets better the more you do it.
Yeah. I know as an entomologist, we're guilty of hanging out traps and then not having the time to actually look at them.
That happens, yeah.
It does. Okay. I'll give everyone kind of a last chance to type in any questions while I let everyone know that our next Pest in Progress webinar will be on Wednesday, November 19th at 12 p.m. Central Time, 1 p.m. Eastern Time. So that's November 19th. Our title is Insect Eavesdropper, a device with highly sensitive microphones that can listen in on insects inside of plants. So that should be interesting. And it will be with Emily Bix from University of Wisconsin, Madison, talking about that. And don't see any other questions about blueberries and spotted-winged Drosophila. So thank you so much, Dr. Isaacs. It was a really useful presentation. I'm glad to see IPM kind of coming back into the system after dealing with spotted-winged Drosophila.
Yeah, we're trying. So thanks for the chance. And yeah, we'll see you all soon.
Yes, thank you all. Have a good rest of your day.